Good morning, everyone. First, I want to thank you for being with me here today. I'm going to start with a brief introduction of SCM Lifescience. SCM, the name SCM has two meanings. One is subfractionation culturing method, which is the most important technology we have developed. And the second one is stem cell medicine. SCM Lifescience produces allogeneic clonal mesenchymal stem cell product. In my talk, I want you to pay attention to the word clonal today. And we have a research center, and we have our own GMP facility. And currently, we are focusing on product for the treatment of immune diseases. I think there are three main hurdles to overcome in the field of mesenchymal stem cells. These include efficacy improvement, disease-oriented treatment, and lowering treatment cost. To improve the efficacy, the strategy we chose was to use clonal mesenchymal stem cells. If you take a look at the bottom of the slide, the final product produced by conventional method are heterogeneous. In contrast, the final product produced by our method are homogeneous population of MSCs. This slide just shows you the conventional isolation method for mesenchymal stem cells. Because of time limit, I'm going to just point out just one thing. People use antibody selection to increase the purity of the final product, but there's no antibody which is 100% specific to MSCs nowadays. So even though you use antibody selection, you still get heterogeneous population of mesenchymal stem cells. To overcome this heterogeneity problem, we developed a new isolation method for mesenchymal stem cells. This method is called subfractionation culturing method. Briefly, we select single colonies of cells from the plate, and we expand them and test them if they have mesenchymal stem cell characters. We only choose the colonies of cells showing mesenchymal stem cell characters, and we move these to we call clonal mesenchymal stem cell library. And using a single colony of cells as a starting material, we establish master cell bank cells and working cell bank cells, and then final product. We have two types of final product, fresh and frozen. This slide shows the real pictures of fresh and frozen clonal MSC product. We have tested the efficacy of our clonal mesenchymal stem cell product in 4 different animal models. And while we tested the efficacy of this clonal mesenchymal stem cell product, we compared with conventional MSC product. The first animal model we tested was GVHD animal model. And as you can see in the figure, the animals treated with our clonal mesenchymal stem cell product shows higher survival rate compared to the animals treated with conventional MSCs. The second animal model was a hair growth model, and as you can see, 3 different clonal MSC product shows much higher efficacy for the hair growth compared to the conventional MSC product. We also found the proliferation of these cells and the expression of some stem cell markers, such as NANOG and KRF4, are higher in clonal MSCs compared to the conventional MSCs. The third animal model was atopic dermatitis model. Although I'm showing you just one representative pictures of animals, the animals treated with our clonal MSC product shows much higher efficacy in these animal models compared to the conventional MSC product or PBS control. This data was confirmed by staining analysis using the sections of tissues. Recently, we also tested erectile dysfunction animal model. As you can see on the right-hand side of the panel, top, the clonal mesenchymal stem cell product shows higher erectile function in this model compared to the non-clonal MSC product. The second order I told you was disease-oriented treatment. And while we characterize these many clonal MSC lines, this is what we found. If we take a look at the panel 2, and when we tested 9 clonal MSC lines, only 5 showed good T-cell suppressive activity. And panel 3 shows when we tested 4 different lines, line 1 shows high level of osteogenic potential, but line 4 did not show any osteogenic differential potential. Line 2 shows good high level of chondrogenic differentiation potential, but line 3 does not. So with this result, result made us to think we could produce disease or tissue-specific clonal MSC lines. For example, you can select subpopulation of clonal MSC lines out of clonally expanded stem cells. For example, if we get some lines showing high level of suppression of immune cells, we can use these cells for the treatment of immune diseases. And if we get some lines showing high level of osteogenic potential, we can use these lines for osteogenic applications. And the third hurdle I told you was lowering treatment cost. The strategy here is we want to increase the productivity of mass culture of MSCs so that we can reduce the cost of treatment. To do this, we tested 7 factors, and out of these 7 factors, we found 3 factors, including oxygen level, antioxidant, and cell density are very important in this mass culture production. And as you can see on the right-hand side of the panel, low cell density and adding antioxidant to this medium really increase the proliferation of these cells. We also tested the combination of 2 or 3 disease factors. We also found there was some synergistic effect using a combination of these factors. And with the data I showed you today, I just want to suggest you that clonal MSC product might be one of the solutions to overcome these main hurdles we're facing these days. And since 2008, we have published more than 30 papers related with clonal MSC lines. And we also established very strong IPs, including isolation, manufacturing, and treatment patterns across the countries. This table shows the list of clinical trials and animal studies we have done and their publications. And this slide shows the pipeline of our company. And we finished phase 1 clinical trial for the treatment of GVHD. We are moving to the second phase 2 clinical trial in a few months. And we are planning to do clinical trials for the treatment of acute pancreatitis, liver cirrhosis, and atopic dermatitis. If you have any questions, please ask me after this section, or you may write an email to me at this address. And thank you for your attention.